Hi there. Today we're going to talk about synovial fluid. This is Body Fluids Lecture, Synovial Fluid Module, and we're going to talk about these following competencies. We're going to talk about performing synovial fluid analysis, and you can see below a list of terms that you will need to know. We're going to talk about how synovial fluid is formed. You should be able to define the function of synovial fluid, to talk about the collection and handling of synovial fluid, and what makes a suitable specimen for analysis. You will also be asked to describe the appearance of normal and abnormal synovial fluids and the tests we use to analyze synovial fluids and the cells that might be seen. Please see the separate slideshow in this unit for synovial fluid slides. You will have a practical in class regarding the slides, separate from your module here. And then you'll be asked to correlate the results with patient diagnosis, treatment, and prognosis. So we're going to look at the appearance of synovial fluids with the possible causes. And then we're going to interpret lab results and classify diseases based on those lab results. So on with the slideshow. Okay, this is chapter 14 in your book. It starts on page 211, so please be sure to read the chapter. So, synovial fluid is secreted by the cells of the synovial membrane, and it's also an ultrafiltrate of plasma. If you remember, serous fluid is also an ultrafiltrate of plasma. Spinal fluid is not. Spinal fluid is made in the spinal column by the ependymal, or in the choroid plexus by the ependymal cells. Now, synovial fluid compared to serous fluid is very viscous, which means it's thick, it doesn't drip real well, it's not as fluid as serous fluid. It does contain high hyaluronic acid and mucopolysaccharides, some plasma protein, and then the glucose and uric acid levels are equivalent to plasma. The hyaluronic acid and the mucopolysaccharides are what make it more viscous. These are unique to this fluid. Then the functions, of course, is supplying nutrients because there's really no blood supply to the joints or in the joint specifically. And then it also provides lubrication of the joints. Joints have to move, just like any other moving part. They need lubrication or things don't work as well as they're supposed to. Then our reasons for analysis. Whoops, excuse me, sorry about that. The reasons we analyze it are to diagnose a possible infection, to see if there's a hemorrhage in there or a bleed. We want to evaluate degenerative disorders such as arthritis, and we want to look at to make sure there is or isn't inflammatory diseases such as lupus or rheumatoid arthritis present. So the procedure that we use to collect synovial fluid is called an arthrocentesis, and they go right into the joint space. You can see a couple examples here of an elbow and a knee. And again, it's a really long needle, and they aspirate that out into a syringe. So very rarely is there enough fluid to put into a bag like a serous fluid. So mostly you will get your specimens in a syringe or tubes. So the tubes that we need then, we'll use a heparin tube, of course, for chemistries and any immunology tests, EDTA tube for hematology, and we'll use a sterile tube for culturing and crystal evaluation. They may also inoculate into broth for culturing. So the things that we look at then, macroscopically, we look at the volume, of course, the color, the clarity. Are there any inclusions? Is it viscous? Does it clot? Is there a mucin clot? We'll talk about that a little more later. Microscopically, then, we'll do a cell count, crystal, cytology. You will also probably do a gram stain. And then chemistries are protein, glucose. We do uric acids, and we'll talk about why. Possibly lactic acid, LDH, and or rheumatoid factors. And then cultures, aerobic and anaerobic. Remember, this is a joint, so there's no air typically present in there, so it's possible for anaerobes to grow. So we classify it according to one of these 4 categories: normal, non-inflammatory, inflammatory, 5 categories, septic, or hemorrhagic. If you look on page 218, it has all the qualities of how to classify these synovial fluids, and you need to be familiar with that. So I thought they had it in here, but they don't. So if a cell fluid is normal, or I'm sorry, a synovial fluid is normal, it'll be colorless to maybe a straw color, and it'll be clear. It might, it won't be hazy at all. So in non-inflammatory, so a degenerative joint disease, it could be yellow, might be slightly cloudy. There'll be a few white cells in it. In a normal fluid, there's usually less than 150 white cells, and they're about a fourth neutrophils. You'll see right around 1,000 white cells in a non-inflammatory joint disease. In an inflammatory joint disease, you're going to see 100,000 white cells, 50% neutrophils. When you get to a septic, that means it's actually due to a microbial infection. You'll see usually 50 to 200,000 white blood cells. Most of them will be neutrophils. The cultures will be positive, okay? And then in hemorrhagic, it'll be due to a traumatic injury, and it'll actually look like blood when you pull it out. You can also have a crystal-induced category where they'll be white and cloudy. 500 to 200,000 white cells, about 90% neutrophils, but you'll also see crystals present, and you won't see a positive culture. So just know that large numbers of white cells, mainly neutrophils, can be due to either crystals or an infection. So again, this is an example of what a normal synovial fluid looks like and an abnormal synovial fluid. Again, we don't use the term xanthochromia. Spinal or synovial fluid by itself is pale yellow typically. So again, xanthochromia is just reserved for cerebral spinal fluid. Now, when a fluid's bloody, it could either be due to blood present in the joint, hemoarthrosis, or it could be due to a traumatic tap. If the physician digs around in there and he goes through a vein or whatever and pulls blood back with it. If it's white or opaque with turbidity, there's either pus or other debris in there. And then when we talk about viscosity, we talk about the string test. All that is you just drop a drop from a pipette or a syringe, you measure it, and it's usually supposed to get longer than 5 centimeters before it breaks. If it breaks before that, then it's a negative or a positive skin string test. I'm so sorry. And then we do the ropes test for a mucin clot, and that talks about how much the hyaluronic acid has polymerized or come together. And if it's good, it's a tight ropey mass. You can see that. If it's poor, the rope clot does not form, or it's really friable, falls apart easily. Again, typically then for cell counts, there should be no red blood cells there. There should be less than 200 white blood cells there. And remember, we have to let that hemocytometer sit longer to allow the cells to settle because it's more of a viscous specimen. So instead of 5 minutes, you may need to let it sit 10 minutes. So if you need to dilute, this is an important point to remember, you need to use saline. If you use any diluent with an acid, such as a Unipet or dilute acetic acid, the sample will clot because the hyaluronic acid will come together. So you have to use saline to dilute these. So then to do a differential, of course, you want to take and spin an aliquot down, get a cell button, and stain it with right stain. And you should see the same type of cells as peripheral circulation. They're going to be in a little bit different numbers. You're going to see primarily monocytes, maybe some macrophages. Lymphocytes will be about 24%, not very many neutrophils. And you'll probably see synovial lining cells. So these are all listed out here. This is a neutrophil. Here is a monocyte / macrophage. There's a lymphocyte, and the synovial lining cells are the big cells with the fried egg appearance. Again, they look similar to mesothelial cells, but we don't call them mesothelial cells because they're synovial lining cells. So mesothelial cells are found in the body cavities, ependymal cells are found in the spinal fluid, and synovial lining cells are specifically found in the joints. You can see LE cells again, the neutrophils with nuclear material engulfed in them, and you may also see TART cells, which are macrophages with nuclear material engulfed in them. Other cells you may see are Reiter cells with the vacuoles. Some malignant cells, you may see organisms. This is a lipophage. This cell is all full of lipids. So then, the thing that's unique to synovial fluid is you may need to analyze that fluid for crystals. Sometimes when people have high uric acid due to gout, they may end up with crystals in their knee joints or toe joints that are very painful. They may be intracellular crystals or they may be extracellular crystals, and they may be due to different causes. So monosodium urate or uric acid are due to gout. Calcium pyrophosphate, CCP, are due to pseudo-gout or fake gout. You may see cholesterol crystals. You may see apatite or calcific arthritis. So there's a change in the cartilage. And you may see drug injections. You may see drug crystals. I think your book has some pretty good pictures of the crystals on page, let me see, page 217. So take a peek at the uric acid versus a calcium pyrophosphate. Usually the monosodium urate or uric acid crystals are real sharp looking, and the calcium pyrophosphate are more square looking. Crystals are very painful, by the way, when they're in a knee or a toe fluid. So chemistry then, total protein. Normal values are 1.07 to 2.13, and if they're high, inflammation or hemorrhage is suspected. Glucose is similar to whatever the current blood level is, and you would suspect that in inflammation or sepsis, then that glucose would be decreased. Lactate may differentiate between septic and inflammatory arthritis, and uric acid is increased in gouty arthritis. They can have gout and you may not see a crystal, so you always need to do that uric acid level. If it's high in a person, it's very likely that they do have gout and just haven't particularly formed the crystals yet. In microbiology, you'll do a gram stain and you'll do acid fast. Sometimes the tubercular or other acid fast bacteria may end up in a joint. And certain organisms like spinal fluids are associated with certain age groups. So in a joint fluid, you will typically see Haemophilus influenza in children. In adults, you may see staph, strep, pneumo, group A beta strep, Neisseria gonorrhea, and in adults over 50, you may see Staph aureus. That being said, you can find other things in synovial fluids. We had a knee fluid one time with a Brucella abortus species in it, which is typically found in cattle and is not typically found in humans. Turns out this guy had been a butcher years and years ago. Apparently he was carrying that Brucella within his body. His knee got inflamed. The infection settled in there. So that was a rare finding, but it's a possibility. So again, we use serology on serum more than on fluids. We may check for autoantibodies, we may check for complement levels. And then again, there's no commercial controls available specific to body fluids, so you will use serum controls for your chemistry. So this table is similar to the table in your book. It helps you classify, but please also pay attention to the crystal category. That can be included in the inflammatory. Some people pull that out into its own category. So again, review of the key points. Synovial fluid is a plasma ultrafiltrate secreted by the synovial membrane. It's more viscous. Its purpose is to lubricate and nourish joints. It can be analyzed for a multitude of reasons: infection, hemorrhage, degenerative, inflammatory diseases. We do it by arthrocentesis. Typically, it doesn't have any red cells and less than 200 white cells. You have to dilute it with saline. You may see peripheral cells in there as well as synovial lining cells. And then we classify the abnormals, the physicians. It's rare that the lab tech will actually say that's inflammatory, that's septic, that's crystal induced, or that's hemorrhagic. Usually the physician does that based on our lab results. So be familiar with the table on page 218. When we talk about polarized light, Polarized light is just a filter applied to the regular microscope, and depending on which way the filter turns, the cells will be either yellow or blue. We don't really get too far into negative and positive birefringent, but you should just associate the words. There's a paragraph on page 216 that talks about negative birefringent crystals. Those are uric acid crystals. They align with the filter and they're yellow, and when they're lying perpendicular, they're blue, okay? The calcium pyrophosphate crystals are positively birefringent, so that means when they're lined up with the compensator, they're blue, and when they're not lined up, they're yellow when they're perpendicular. And so when you have those polarized scopes, it will tell you which way the filter is going, and so you can tell whether they're negative or positive birefringent. But just remember that negative birefringents are unique to uric acid crystals. They're yellow when lined up and blue when not lined up. And then positive birefringent are associated with calcium pyrophosphate, which are pseudogout, and they are blue when lined up and yellow when not lined up. So if you need to make yourself a flashcard for that, you might want to do that. This is the end of our lecture. Please review the slideshow, and we will see you later.